Hello everyone, welcome back to our channel Indian Pharma Guide. In our last video, we discussed about the definition, history and importance and potential health benefits of the probiotics, and today I am going to explain you about the mode of action, identification, advantages and disadvantages and finally applications of the probiotics. In this first one is mode of action. So probiotics are the live organisms that can improve the health of the body by restoring the balance of the good bacteria. So they work in a number of ways, that is competitive exclusion, antimicrobial production, barrier function, immune regulation, energy production, enzymatic activity, inflammation reduction. In this first one, competitive exclusion. So here probiotics compete with the pathogens for nutrients and receptors, so making it difficult for pathogens to survive in our body. Second one is antimicrobial production. So here probiotics produce the substances like organic acid and hydrogen peroxide that inhibit the growth of the pathogens. Next one, barrier function. So here probiotics strengthen the intestinal barrier by increasing the mucus production and the expression of the tight junction protein. Next one, immune regulation. So here probiotics modulate the immune response by increasing the activity of the T cells. Next one, energy production. Probiotics produce the short chain fatty acids, vitamins, amino acids and secondary bile acids, so that it will increase the production of energy. Next one, enzymatic activity. So here probiotics metabolize the toxic substances and carcinogens. Last one, inflammation reduction. So here probiotics reduce the inflammation by inhibiting the TLR2 and TLR4 and by stimulating anti-inflammatory mediators. So finally probiotics are available as the foods or the supplements. Next one, identification. Probiotics can be identified by their morphology, biochemical features and genetic makeup. First one, morphology. In morphology, probiotics can be identified by two ways. One is the microscopic examination, second one colony morphology. In microscopic examination, shape, size and other characteristics of the cells will be observed. And in colony morphology, shape, size, color and other characteristics of the colonies has been observed. Second one, biochemical features. In this first one is growth. Determine the optimal growth temperature and how the microorganism grows in the liquid and solid substrates. Next, second one is the production. So here it will determine if the microorganisms produces a certain compounds such as dyes, hydrogen sulfide or ammonia. Next, last and final one is resistance. So here it will determine if the microorganisms is resistant to antibiotics, bacteriophages or other drugs. Last one, genetic makeup. So in this 16S rDNA sequence analysis, so here 16S rDNA gene will be used to identify the bacterial species. Next one, matrix assisted laser desorption or ionization time of flight mass spectrometry. So here in this method is used to identify the genera that produce the lactic acid. Last one, molecular techniques. So here some molecular techniques will be used like polymerase chain reaction, reverse transcriptase PCR or real-time quantitative polymerase chain reaction to count the bacterial cells. Next, advantages and disadvantages of probiotics. So here probiotics are the living organisms that can be used to treat and prevent the diseases, but that can also affect the efficacy of the other drugs. Next one, advantages. So here probiotics are used to treat and prevent the diseases and maintain the gut health and modulate the immune system and finally co-adjuvants in the therapy. This first one, treat and prevent diseases. So here probiotics can help to treat and prevent a variety of diseases including digestive disorders, inflammatory diseases and some cancers. So second one, maintain the gut health. So probiotics can help to maintain a healthy balance of the gut bacteria. So next one, modulate the immune system. So here probiotics can help to modulate the immune system, which can help to reduce the inflammation. So last one, co-adjuvants in the therapy. Probiotics can be used as adjuvants in the therapy for several ill patients. Next one, disadvantages of probiotics. In this first one, biological and biopharmaceutical barriers. Probiotics can lose their activity in the stomach or in the presence of the bile salts, so they can also lose their visibility during the preparation and the storage. Second one, antibiotic resistance. So some probiotic bacteria may be resistant to the antibiotics, which can lead to life-threatening conditions. Next one, can affect the drug bioavailability. So here probiotics can directly or indirectly affect the bioavailability, efficacy and safety of other drugs. Last one, can cause the temporary diarrhea or bloating. So probiotics can produce the short chain fatty acids or gases in the gut, which can cause the temporary diarrhea or bloating. Last and final one, applications of the probiotics. So probiotics can increase the microbial richness and diversity, and it increases the enzyme production, improve the immune microenvironment and improves the intestinal permeability. So in this way probiotics can alleviate intestinal diseases too. And that's all about the probiotics, and thanks for watching, and please do subscribe for our channel for more videos.